Good morning, everyone, and welcome. I am Dr. S.K. Singh, and I'm glad to have you here today. Today, we'll be exploring the topic of calculations of uniformity of content. This is the first slide, so I just want to give you a quick overview of what we'll cover. We'll look into the importance of content uniformity, some key calculation methods, and how these are applied in practice. So, let's get started and dive into the fascinating world of content uniformity calculations. Content uniformity is a crucial quality control test that we use to make sure that every single tablet or capsule in a batch contains the correct amount of the active pharmaceutical ingredient, or API. It's all about confirming dose consistency, so patients receive the right amount each time. When we talk about uniformity of content, especially using HPLC or high-performance liquid chromatography, we're employing a precise analytical method to measure the API in each dosage form. This process is key because it helps us ensure product quality and safety, making sure that no pill is under or overdosed. Ultimately, content uniformity ties directly into the overall goal of quality assurance in pharmaceuticals, delivering safe, effective medications that meet strict regulatory standards. Now, let's look at the acceptance limits for the uniformity of content in single-dose preparations of the IP. When preparing solutions according to the monograph, we typically start with a blank preparation, which serves as our baseline. Then, we have reference solutions or working standards, which are prepared in diluents or mobile phases. These standards are crucial because they help us compare and ensure accuracy. Next, we prepare the test solution, which involves taking one tablet or capsule and dissolving it in the chosen diluents. The purpose of this test is to check whether each individual unit contains an active substance within the acceptable range, based on the average content of the sample. This uniformity test is particularly applicable to tablets or capsules containing 10 milligrams or less than 10 milligrams of active ingredient, or less than 10% WW. It's important to note, however, that this test does not apply to multivitamins or products containing trace elements, since their content isn't suitable for this kind of analysis. Moving on to how we analyze this, the uniformity of content is often assessed by HPLC, or high-performance liquid chromatography. HPLC provides a reliable, precise way to measure the individual contents of active substances in each unit, ensuring they fall within the specified limits. Overall, the goal here is to ensure each dose delivers the intended therapeutic effect, maintaining quality and consistency across all units. Let's walk through the sequence arrangement for this part of the process. First, we start with a blank, which essentially sets a baseline before we introduce our reference solution. This reference solution or standard preparation involves 5 injections, and these are crucial because they serve as our benchmark for comparison throughout the analysis. Next, we move into preparing our samples. We've got 10 different sample preparations, labeled from 1 to 10. These are the actual test samples we're analyzing, and preparing them carefully ensures that our results are accurate and reproducible. After we've prepared all our samples, we include a reference solution again, this time for bracketing purposes. Bracketing with a reference helps us account for any variability in the HPLC system during analysis, maintaining consistency across runs. Now, the key point here is the concept of the uniformity of content by HPLC. This step is fundamental because it verifies that each sample contains a consistent amount of the active compound. By analyzing all these preparations, we can confirm whether our manufacturing process produces uniform results, which is essential for quality control and compliance. Connecting all these points to the overall theme, this sequence arrangement and the uniformity check are vital steps in ensuring the reliability and accuracy of our analytical method. They help us validate that our samples are consistent, our method is robust, and our results are trustworthy. Let's now look at some important system suitability observations that we need to consider. These include parameters like asymmetry, theoretical plates, and relative standard deviation, or RSD, or any other criteria specified by the method or monograph. These factors help us ensure that our chromatographic system is performing properly before we analyze samples. Focusing on uniformity of content by HPLC. This is a key aspect because it verifies that the method consistently produces accurate and precise measurements across multiple samples. In practice, we check that the system's performance parameters meet the specified criteria, which ensures the reliability of our results. Remember, maintaining these standards is critical for the overall integrity of our analytical method and for compliance with regulatory guidelines. So, by observing asymmetry, theoretical plates, and RSD, along with verifying content uniformity, we uphold the robustness and accuracy of our HPLC analyses. Now, let's take a closer look at the content of primaquine phosphate tablets. These tablets are expected to contain not less than 90% and not more than 110% of the stated amount of primaquine, which is chemically known as C15H21N3O. The usual strengths available are 7.5 mg and 15 mg per tablet. It's important to note that 13 mg of primaquine phosphate roughly corresponds to 7.5 mg of the active primaquine base, highlighting the conversion we need to consider during analysis. To determine the actual content, we use liquid chromatography, as described in the assay method. The process involves preparing a test solution by powdering one tablet, dissolving it in 20 ml of mobile phase with ultrasound for about 3 minutes, and then diluting the solution up to 50 ml with the mobile phase before filtering. For the reference solution, we dissolve a weighed amount of primaquine phosphate reference, standard in the mobile phase, and dilute it to match the expected concentration of the test solution. We then inject both the test and reference solutions into the chromatograph and analyze the results. By comparing these, we can accurately calculate the amount of C15H21N3O present in the tablet. This approach ensures the uniformity of content, which is essential for quality control and consistent therapeutic effect. It's a fundamental part of ensuring the safety and efficacy of primaquine tablets. Now, let's take a closer look at the primaquine tablets and how we assess their quality. One key aspect is the uniformity of content, which is crucial for ensuring each tablet delivers the correct dose. To evaluate this, we use high-performance liquid chromatography, or HPLC, a highly precise analytical technique. HPLC allows us to measure the amount of active ingredient in each tablet accurately and consistently. This step is vital because it helps us confirm that the manufacturing process is reliable and that patients receive a safe and effective dose every time. Overall, maintaining uniformity through methods like HPLC supports the integrity of our medication and our commitment to quality assurance throughout the production process. Now, let's take a closer look at the primaquine tablets and how we assess their quality through the uniformity of content. Here, we use high-performance liquid chromatography, or HPLC, to ensure each tablet contains the correct amount of active ingredient. This method is highly precise and sensitive, allowing us to detect even small variations between tablets. The key point is that uniformity of content is crucial for both efficacy and safety. Patients need to get the right dose every time, and we want to prevent any under- or overdosing. By applying HPLC, we can confidently verify that each batch meets the strict quality standards. This step is an essential part of the overall quality control process, helping us maintain consistency across production runs. It ties into our broader theme of ensuring the safety, effectiveness, and reliability of the medication we deliver. For further support, please don't hesitate to reach out. If you have any questions or need additional information, we're here to help. You can contact us through the provided details. Thank you so much for your attention and engagement today. We truly appreciate your time and interest. Remember, we're always available to assist you as you apply what you've learned. Thanks again, and have a great day.